Hello everyone, and welcome to today's live broadcast, Acoustic Experience Alters How You See the World, presented by Dr. Stephen Lomber, a Canada Research Chair in Brain Plasticity and Development and a Professor of Physiology, Pharmacology, and Psychology at University of Western Ontario. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is a leading scientific social networking website and producer of educational and virtual events and webinars. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on that screen. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble hearing or seeing the presentation, click on the Help Desk button located in the promotional board at the bottom center of the screen, or use that Ask a Question box to let us know you're having a problem. Also, this presentation is educational and thus offers continuing education credits. Click on the Continuing Education Credits tab that's located on the top right corner of the presentation window and follow the process for obtaining your credits. Without further ado, please join me in welcoming our presenter today, Dr. Stephen Lomber. I will now turn the presentation over to him. Welcome, sir.
Great. Thank you very much. Today we'll be talking a little bit about brain organization and cortical plasticity, and we'll explain a little bit about the work we do here in our lab. I'd like to start with an overview of the cerebral cortex, its organization, and some of the plasticity work we understand already. So what you see here is a dorsal view of the human brain, basically looking down from the top, and this is kind of the classic view that many of us think of when we think of the human cerebral cortex. But unfortunately, you're really only looking at a small portion of the cortex when you look at the brain from this view. Very little of the cerebral cortex is actually on the surface of the brain. The vast majority of the cerebral cortex is actually buried deep down into the sulci that you can see on this horizontal section. So what you see on the right-hand side is a dorsal or a top view of the brain that's been cut along the red line on the left-hand side of the slide. And the top of the brain has been lifted off, and you can actually see down into the substance of the brain itself. So what you're looking at here is mainly gray matter of the cerebral cortex and underlying white matter. And you can see how the thin gray line, or the thin kind of covering, is the gray matter of the cerebral cortex, and in the human brain is only about 2 to 3 millimeters thick. But you can see how deep it penetrates into the brain. If we use the estimates of Ted Jones and Alan Peters and can actually roll out the entire human cerebral cortex to make it into a thin sheet, you'd actually find that the entire size of the human cerebral cortex is about the size of a small dining room table. So it's a very large sheet, a very large thin sheet of neurons that's compacted into a very small space because it's kind of folded up like a radiator inside the human head. Another important thing we know about the cerebral cortex is its functional specialization. Over time, we've learned that different sensory, motor, and cognitive functions are localized to different parts of the human brain, such that if you look towards the right-hand side of the slide, areas 17 and 18 would be where primary visual cortex is localized, or area 41 towards the center is where primary auditory cortex is localized, or towards the top of the slide, areas 3, 1, and 2 is where primary somatosensory, or touch cortex, is localized. As neuroscientists, this affords us a great opportunity to be able to look for particular functions in particular parts of the brain. But it also has a downside, and that is we know that focal regions of damage can have devastating functional consequences. So a stroke that's localized over a region of visual cortex or localized over a region of motor cortex can have profound visual or motor impairments per se. Today we're going to be talking about cortical plasticity, and the definition I'll be using for cortical plasticity is that it's changes that occur in the function and organization of the cerebral cortex as a consequence of experience. Now, experience can be a very broad word. Often you can think of an experience as learning a second language or learning to play a new sport. Today we'll be using it in the term of the presence or absence of a particular sense. So we'll be talking about it in terms of the presence of hearing or the absence of hearing, such as deafness, or the presence of vision or the absence of vision, such as blindness. Much that we've learned about cortical plasticity was based upon the pioneering work of David Hubel and Torsten Wiesel, who were examining visual cortex in cats and monkeys. In this slide, you can see some of their seminal anatomical work, where they injected radioactive amino acids, in this case proline, into the eye of a monkey. So you can see the monkey brain in the upper left-hand corner with a kind of a syringe as if it's going into the right eye. The radioactive substance was then taken up by the ganglion cells in the eye and transported anterograde to the LGN, which you can see as a Nissl stain section on the right-hand side of the slide. The radioactive amino acid was then taken up by the neurons that reside in the LGN and then transported up to primary visual cortex, which you kind of see illustrated on the top of the slide. When they processed visual cortex for the presence of this label, and particularly in layer 4 of visual cortex, what they found is what you see in the upper right-hand side, this kind of zebra stripe pattern of labeling in the visual cortex, where regions that appear white were regions that were in receipt of projections from the injected eye, and the regions that appear darker or black were regions that were in receipt of projections from the non-injected eye. So you can see a kind of a loose 50-50 distribution in the presence of the tracer in visual cortex, and how it kind of alternates black, white, and so forth. This was a really compelling finding in its own, but then they took it a step further and wanted to look at how modifiable these projections might be. So what they did with some newborn monkeys was to suture one eyelid closed and then let the animal develop for some period of time, basically only seeing through one eye. And then they applied the exact same tracer to the open or the good eye, and when they did that, they found the pattern of labeling that you see in the lower section on the right-hand side, where now the visual cortex is almost completely in receipt of projections from the good or the open eye. And in fact, there are very few projections from the closed or the bad eye. So this really showed a profound demonstration of how plastic the anatomical projections are in the visual system in a young primate. Then they obviously took the next step and then reopened the closed eye and found that the pattern would go back to what you see in the top section after a period of time. And you could actually go back and forth modifying these projections until you reached a particular time in development, which then became known as a critical period. Mainly being that before this critical period or time in development, the projections were modifiable, but then once you reach the end of this developmental window, basically you could think of the projections were locked in for the remainder of the animal's life. And these experiments launched literally 1,000 studies in ocular dominance plasticity, as it's known. But there was a downside of these experiments. They also led us to believe that this kind of plasticity was only afforded early in development. There wasn't any sort of cortical plasticity later on. Obviously, over time, we found this isn't to be true, but these experiments are extremely important nonetheless. A little bit more recent from those studies was some work done in Mike Merznick's lab at the University of California in San Francisco, where he was examining the brains of adolescent owl monkeys. So at the top of this picture, you can see the brain of an owl monkey, and he was specifically looking at somatosensory cortex. So you can see here where the representation of the body and the face would be on the cerebral cortex of an owl monkey, and they mapped out the representation of the 5 fingers of the owl monkey that you can see in the bottom left. And on the right-hand side, you can actually see how those 5 digits were represented in the cerebral cortex, digits 1 through 5, and it almost looks like a hand laid over the surface of the brain. So they identified where the representation of the 5 digits are in somatosensory cortex, and then they asked the simple question of what would happen following amputation, or what happens to the representation of the hand if 1 digit went missing. So they did this experiment, and what they found is in the upper left, you can see the animal with its digits 1, 2, 4, and 5, but with a 3rd digit missing. And after several months, they went and recorded from somatosensory cortex from this animal, and what they found was that there wasn't a region that was no longer responsive to tactile stimuli. In fact, what had happened was the representation from the second and the fourth digit actually expanded into the territory that would previously occupy the representation of the third digit. And this actually correlated with enhanced sensitivity in the second and fourth digit, presumably because there was now more cortical territory dedicated to processing that information. The other interesting thing you'll see, though, is overall the hand representation didn't get any larger. It was pretty much the same size as the hand representation as before. It's just that there was now more cortical territory dedicated to processing information from the second and the fourth digit. Even more recently was work from Josef Rauschecker's lab. This work is a little bit different from the previous two studies we were just talking about. In the previous two studies, we were looking at competition within the visual system and competition within the somatosensory system. In this study, we're actually looking at competition between sensory systems. So here you can see in the upper picture is an outline drawing of the cat cerebral cortex. And there's one area that's kind of highlighted with the yellow, blue, and red. This is the anterior ectosylvian sulcus. And in the lower two panels, on the left is an intact animal, and on the right is this region, a blind animal. So what you see under the intact is, what's unique about this area is that you have three relatively unimodal sensory fields living in close proximity to each other. So in yellow, you have a somatosensory region, in blue is an auditory region, and in red is a visual region. In these studies, what the investigator had done was to do bilateral eyelid suture and have the animals live for some period of time without receiving any normal visual input. And what was found was that the normally visually responsive region shown in red in the left panel now became acoustically responsive as shown in the right panel. And this was actually correlated with an increased auditory spatial sensitivity found in these animals, and that they were much better at localizing acoustic stimuli than they were, or what can be found in a normal intact animal. And this has led to the idea of what we refer to as adaptive cross-modal plasticity. And anecdotally, you probably know of this phenomenon with things like, or individuals such as the blind piano tuner, who has an exquisite sense of pitch and has acoustic abilities that are much greater than an individual who's normally sighted. But the idea is that following the impairment of a sensory system, such as blindness or deafness, it is the enhanced perceptual performance identified in the remaining systems, whereby one or more modalities compensates for the loss of another modality. Another example of this was the work done from Franco Lapore's lab at the University of Montreal, where they studied enhanced auditory spatial localization in the blind. Very similar to the cat work we just saw previously from the Rauschecker lab. In our own lab, we've been studying audition and vision in cats as well. Here you can see a picture of the auditory cortex of a cat. And this large area in yellow is what would normally be auditory cortex in a hearing animal, and the area outlined in orange is what normally would be visual cortex. So this is how, under conditions of normal development, the cerebral cortex was set up. But the question we've been asking is, absent normal hearing, such as in a congenitally deaf animal, what actually happens to this underutilized region of auditory cortex? Since it's no longer in receipt of auditory projections, do other modalities or other senses, such as vision and somatosensation, exploit this underutilized area to enhance the perceptual abilities of, say, vision or somatosensation? Okay, about 10 years ago, a really fantastic review article came out by Daphne Bavelier and her colleagues, which appeared in Trends in Cognitive Science, and asked the simple question, do deaf individuals see better? And what this was a meta-analysis of all of the human psychophysical literature done in congenitally deaf subjects up to that point. And what she found was that you don't have a general enhancement in visual abilities in the deaf. What you have are enhancement of specific visual abilities. And she found that there were very profound findings or improvements in visual orienting and reorienting, in visual motion processing, and in visual stimulus onset detection. And this was an important review paper for us because it helped guide our own studies into this topic. So the 3 points I'd like to make with you today, and hopefully you'll remember, are first off, is that congenitally deaf cats have specific visual abilities that are superior to hearing cats. Right? It's not going to be a general improvement, it's going to be specific visual abilities as outlined by the Bavelier study. Second, regions of deaf auditory cortex, that is, regions of the brain that would normally be auditory cortex in a hearing animal, are responsible for mediating the enhanced visual abilities of the congenitally deaf. And finally, the cross-modal reorganization of the congenitally deaf switches the sensory but not the behavioral roles of auditory cortex. We'll look at this more specifically in a little bit. Okay, so what this picture is a cat brain, kind of shown in the foreground. So you can see the cerebral cortex, the cerebellum behind that, and the spinal cord behind that, superimposed on a cat skull, superimposed on a cat head. So you can see kind of relative to each other where the cat cerebral cortex in the brain in its total actually resides in the head of a cat. The animal we're specifically going to be talking about today is the congenitally deaf cat, otherwise known as the deaf white cat. These cats have a very characteristic phenotype and are special compared to most cats. They're going to have white fur, either long hair, as shown in the upper left-hand corner, or short hair in the lower right. But what usually tends to make them quite unique is that they have two blue eyes. So if you have a cat that has white fur and two blue eyes, there's about a 75 to 80% chance that it was born deaf and has never heard. We obviously confirm this with auditory brainstem responses shortly after the animals are born. All of our animals are derived from a breeding colony in Hanover, Germany, directed by Andre Kroll, who's in the picture with myself in the upper right-hand corner. A couple of important things to point out about these animals are, first off, they don't have any Siamese traits or albino traits. They actually have very normal visual systems. Both Siamese and albino cats have unusual visual systems, whereas these cats just have very low levels of melatonin. And they can actually be born just deaf in one ear. It can be a case where an animal can be born with one blue eye and will hear in the ear ipsilateral to the blue eye, or on the same side. And if they have a brown eye on the other side, they may actually be able to hear through the ear that's ipsilateral to the brown eye. So they can be both bilaterally deaf, as you can see with the animals here that have two blue eyes, but they also can be born with one brown eye and one blue eye. Okay, so the first set of experiments we're going to talk about will be examining dorsal stream functions following deafness. And streams in cortex were first established in visual cortex, the dorsal stream being more spatial in nature and the ventral stream being more perceptual in nature, the so-called where and what pathways. So we're going to consider where type functions in the first set of experiments and what type functions in the second set of experiments. So the two aims that we're going to look at are, first, to determine what dorsal stream visual abilities may actually be enhanced as a consequence of deafness, and second, to determine what role deaf auditory cortex may play in mediating these superior visual functions. So basically, the progress for these studies are the white cats and hearing cats are born. Deafness and hearing is then confirmed with auditory brainstem responses at 1 month of age to establish the presence of hearing or absence of hearing. After reaching maturity at about 1 year of age, we're going to start training and testing on a battery of visual tasks. Then we're going to implant cooling loops over desired regions of deaf or hearing auditory cortex to permit their focal reversible deactivation. We'll see that precisely in a little bit. And then we're going to test all cats on all tasks during reversible deactivation of each cortical locus. At the end, we perform acute cortical recordings electrophysiologically and pathway tracer injections to look at anatomical rewiring, and then perfuse and process the tissue. Okay, so the first aim is to determine what dorsal stream visual abilities may be enhanced as a consequence of deafness. So to do this, we're going to psychophysically test both deaf and hearing cats on a battery of visual tasks to ascertain what visual abilities are actually enhanced as a consequence of deafness. In this study, we're going to look at 7 different psychophysical tasks: vernier acuity and grading acuity to look at sharpness of vision, orientation discrimination, the direction of visual motion, velocity of motion discrimination, the detection of movement, and detection across the visual field. So to cut to the chase, of all these 7 tasks, most were the same between both the hearing and the deaf populations, but the 2 that were different were the detection of movement and detection across the visual field. For these 2 tasks, the deaf animals had an advantage or superiority to the hearing animals. So let's look at those 2 tasks specifically. First, detection across the visual field. In the left-hand side, you'll see an arena with an animal standing in the center. The animal's attention is drawn by the illumination of a red LED at the 0 position. and then that is extinguished, and another red LED is flashed somewhere at one of the different 15-degree positions around the arena. In this case, you can kind of see how right 75 is indicated to have flashed. The animal then orients towards it, approaches it, and receives a food reward. What you see on the left-hand side is a polar plot, kind of looking down upon the apparatus from above. So you can imagine the cat's head at the 0% correct position. And the length of each green line indicates percent correct for the hearing cats for the different spatial positions. And the red extensions show how much better the deaf cats did in comparison to the hearing animals. So you really see out in the far periphery, 60, 75, 90 degrees on both sides, that the deaf cats were significantly better. Since we didn't find any difference between the left or the right sides, if we collapse across and now look at just eccentricity with from 0 to 90, the height of the green bars are percent correct of the different spatial positions for the hearing cats, and the height of the red bars are the abilities or the percent correct of the deaf animals. So here you can see for the central positions, there was no difference between the hearing and the deaf populations. But if you went out to the far peripheral positions, 60, 75, 90 degrees, you can see how much better the deaf animals did. And the other task, detection of movement, in this case, the animals are in a 2 alternative force choice apparatus where they stick their head into a small box and their 2 nose press keys, one in front of a left stimulus, one in front of a right stimulus, and they see a food reward from below when they make the correct choice. In this experiment, they had to select, or their 2 options were a field of stationary dots and a field of coherently moving very slow dots. I mean, very, very slow. And their task was to identify which field was actually in motion. And this was done through a staircase procedure where basically as they did better and better, the dots would move slower and slower until we could actually determine what the threshold or what their best performance was. And in this case, we found that best performance for the hearing animals was around 1.3 to 1.4 degrees a second. But the deaf cats actually had much lower thresholds at down around 0.5 degrees a second. So the deaf animals could actually detect much slower speeds than the hearing cats could, which would obviously be a performance advantage. Okay, so to summarize to this point, the congenitally deaf cats had visual abilities that were similar to hearing cats on the vast majority of the tasks that we examined. However, the congenital cats had visual abilities that were superior to the hearing cats, specifically on the detection of movement and visual detection in the peripheral field. Now, in the second part of this study, we're actually going to look at where these visual abilities may actually be taking place. So in aim 2, we want to determine what role deaf auditory cortex may play in mediating these superior visual functions. So to do that, we're going to deactivate specific sites in deaf auditory cortex while the deaf cats perform the visual tasks for which they possess these enhanced visual abilities. Okay, so in this study, we're going to examine 4 different areas in the presumptive dorsal stream of auditory cortex. A primary auditory cortex, shown as A1, the anterior auditory field, AAF, the posterior auditory field, PAF, and the dorsal zone indicated as DZ. So to do this, rather than taking a traditional lesion approach and having to use large numbers of animals, we actually use a reversible deactivation technique that we've championed in our own lab, or the Cryoloop Technique. This can originally be found in a 1999 Journal of Neuroscience Methods paper. It's quite simple. Basically, a methanol is drawn from a reservoir and then pumped through a methanol and dry ice bath that's around minus 80 degrees Celsius. It's then exported to the device that's implanted over a discrete region of the cerebral cortex, where it then cools the brain, and then the methanol is returned. Basically, what you're doing is inducing a small region of focal hypothermia, basically chilling a small region of the brain, turning the cells off. But the interesting thing isn't that you can turn the cells off. The interesting thing is when you stop cooling, the neurons begin firing like normal again. So you're basically rendering a region of the brain silent and then turning it back on. On the left-hand side, you can actually see one of these devices that have been fabricated for placement over a particular region of the brain. Hypodermic stainless steel tubing is shaped to conform to a particular region of the brain. And when you see the arrow pointing at loop, that would be the region in contact with the cortical surface. You can also see a copper-colored wire leading up to the blue plug, which is where the thermometer would plug in and monitor the temperature at the point called TC, or thermocouple. And if you look at the top, you can see the inflow and outflow of the methanol, and then a protective cap that goes over the whole thing, basically protect those two tubes when the animal isn't actually being tested. Now, there are a couple things you need to know about reversible cooling deactivation. First off, tissue temperatures below 20 degrees Celsius eliminate synaptic transmission. So if you deactivate any region of the brain to below 20 degrees Celsius, you'll eliminate synaptic transmission. However, you don't impair underlying axonal transmission, mainly because the myelin over axons really protects them or shields them from any sort of cooling. So you might actually turn off the neurons in an area that you're interested in, but you won't interfere with underlying fibers of passage. The deactivation is highly localized. During the whole time you're performing a cooling deactivation, warm blood is being pumped in, nutrients and so forth. So the cooling doesn't actually spread very far at all, generally only 1 to 1.5 millimeters away from the loop. Deactivation can be induced or reversed within minutes, which is really beneficial, particularly for behavioral studies. Each animal serves as its own control. So from each animal, you can actually deactivate multiple cortical sites, but you can also get both control and experimental data from each animal. So this dramatically reduces the animals that you actually need to train or have participate in your studies. And finally, for us, multiple cortical sites can be examined each animal, so we can actually examine more than one area per animal. In this case, in these studies, we're going to examine 4 different areas in each animal. Okay, what you see here are the sites of 4 different loops in the upper left-hand panel, and then the temperature recordings that were taken at over 300 sites across auditory cortex, which were recorded basically with a device that looks like just like an electrode, but is basically used for temperature monitoring. So here you can see the actual temperature range on the scale on the right-hand side. And once again, the important thing to know is where 20 degrees Celsius is. So from light to dark blue, you can actually see these would be the regions that are deactivated. For the anterior auditory field, you can see how well localized the region of cooling is. For A1, you can see how well localized the region of cooling is. And then also for the dorsal zone in panel E, and then finally for the posterior auditory field. What's interesting about the loop that was in the posterior auditory field is it actually descends down into the sulcus and had a heat shielding compound applied to its anterior side. So in this case, the cooling was only on the posterior bank of the sulcus and not on the anterior bank of the sulcus. Okay, so now let's look at performance with our tasks. So in this case, we're looking at detection across the visual field. And the first thing we're going to do is deactivate all 4 sites simultaneously. So we're going to see if there's any sort of effect on performance when we deactivate all 4 of these fields at the same time. So this seemed like the prudent thing to do before actually doing individual deactivations. So in this case, you can see the height of the red bars are performance of the deaf animals, and the height of the blue bars are performance of the deaf animals while all 4 areas were simultaneously deactivated or cooled. That's why they're shown in blue. So what you see here is there's no effect on the central positions, but on those 3 peripheral positions, 60, 75, 90 degrees, you can see how performance drops when all 4 areas are simultaneously deactivated. This then basically gave us permission to go and do the individual deactivations of each area. So here you can see on the top panel the bilateral individual deactivation of the posterior auditory field. Down from that is the dorsal zone, below that A1, and finally at the bottom the anterior auditory field. And what you see is there was no change in performance for 3 of the 4 areas, but for the top panel, for the posterior auditory field, you can see that there's a significant drop in performance for the 3 peripheral positions. And in fact, the entire deficit we saw during deactivation of all 4 areas could be attributed to just the deactivation of the posterior auditory field. But the neat thing is when we do the comparison. So here you can see the results of bilateral deactivation of the posterior auditory field in comparison to the other results. So here, the green bars indicate performance of the hearing animals, the red bars indicate performance of the deaf animals, and the blue bars indicate performance of the deaf animals during bilateral deactivation of the posterior auditory field. So what you can see is the excellent or enhanced performance of the deaf animals and how this enhancement was removed when the posterior auditory field was bilaterally deactivated. And in fact, the entire advantage the deaf animals had was eliminated during bilateral deactivation of the posterior auditory field. Performance didn't get any worse than normal hearing animals, as you would expect, but the performance advantage was completely eliminated. Now, an interesting question to ask is, why the posterior auditory field? What is unique about the posterior auditory field, or what do we already know? Well, from previous work that we've done in the lab, in hearing animals, we've found that bilateral deactivation of the posterior auditory field has a profound impairment in sound localization, or the localization of an acoustic stimulus. So if you look at the middle bottom panel, you can see how poorly the animals do during bilateral deactivation of the posterior auditory field in comparison to normal performance in the upper three panels. So in hearing animals, the posterior auditory field makes a significant contribution to auditory localization, but in this population of congenitally deaf animals, what we find is the posterior auditory field is making an important contribution to enhanced visual localization abilities. Now, what about the other task, the detection of movement? Once again, we're going to deactivate all four areas simultaneously while the animals perform this discrimination. And here what we found is the red bar is performance of the deaf animals, and the blue bar is performance of the deaf animals when all four areas were simultaneously deactivated. Once again, you can see the threshold goes up, or performance gets worse in these animals when these four areas are all deactivated at the same time. Now let's look at what happens when you look at individual deactivations of each four of these areas. In the first panel, in A, you can see that there is no difference in performance when the posterior auditory field was deactivated bilaterally, which is in contrast to the previous task we just looked at, where the posterior auditory field was so important. We also don't see any change in performance during deactivation of A1 in the lower left or AAF in the lower right. However, you do see a big change in performance during deactivation of the dorsal zone, or area DZ. In fact, what you'll see is performance gets worse, or threshold goes up, during bilateral deactivation of the dorsal zone. So here you can see that in the deaf animals, performance gets worse following bilateral deactivation of the dorsal zone. But let's look at the comparison. Once again, performance of the hearing animals is shown in the green bar, and performance in the deaf animals is shown in the height of the red bar. And the height of the blue bar shows performance of the deaf animals while the dorsal zone was bilaterally deactivated. So what you can see is that the advantage of the deaf animals went away while the dorsal zone is bilaterally deactivated, and performance is now no different from that of the hearing animals. So once again, what's so special about the dorsal zone? Well, at the time we conducted these experiments, we didn't actually know much about what the dorsal zone contributes to auditory behavior, but since then have done some experiments looking at the direction of acoustic motion, and found that there are profound deficits in the direction of acoustic motion discrimination when you actually bilaterally deactivate the dorsal zone. So in fact, the dorsal zone actually contributes to acoustic motion processing in hearing animals, but in the deaf animals is actually contributing to visual motion discrimination, or localization, visual motion processing. So let's summarize this first study. Here you can actually see that we found that there were two tasks where performance was enhanced in the congenitally deaf animals. That was visual detection of the peripheral field and the detection of movement. And when we deactivated the posterior auditory field bilaterally, we got a profound deficit on the visual detection of the peripheral field, but not in the detection of movement. In contrast, when we deactivated the dorsal zone bilaterally, we found a deficit in the detection of movement and no deficit in the visual detection of the peripheral field. This is classically known as a double dissociation, where you show that one area contributes to a task and not the other, and vice versa. So here we're actually finding that there's a visual double dissociation in what would normally be auditory cortex. So this region of the brain normally doesn't contribute to any of these functions, but following the loss of normal hearing input is now contributing or making a key contribution to visual functions. This led us to develop what has now been referred to as the supramodal hypothesis. and that is the cortical areas that have been physiologically reorganized in response to a sensory loss, such as blindness or deafness, are involved in behaviors that are similar to those of hearing in sighted subjects, but are mediated by the replacement modality. Okay, so using the example of the posterior auditory field, in hearing animals, the posterior auditory field normally is involved in auditory localization, but following hearing loss, in the deaf animals, it's now involved in visual localization. In both cases is involved in localization. It really depends on whether audition is present or absent. Okay, in the second group of studies, we actually want to look at ventral stream functions following deafness. So these functions are going to be slightly more cognitive in nature. In this case, we actually want to determine if the learning or recall of any visual discriminations are enhanced as a consequence of deafness, and then once again determine what role deaf auditory cortex may play in mediating superior cognitive functions. So to do this, we're going to look at a different set of 4 areas. These are areas of presumptive ventral or what stream in auditory cortex. So this stream emanates from A1 and then passes down to A2 and then into the temporal area, abbreviated T, or insular area, abbreviated IN. This is the presumptive what pathway or ventral stream in auditory cortex. So the first aim is to determine if the learning or recall of any visual discriminations are enhanced as a consequence of deafness. To do this, we're going to test both deaf and hearing cats on a battery of visual discriminations to ascertain if the learning or recall of any visual discriminations are actually enhanced as a consequence of deafness. So in this case, all the testing is going to be done in 2 alternative force choice apparatus, like you see pictured here, where the animal is presented with one stimulus on one screen and a different stimulus on another, and they're going to indicate which is the rewarded or the preferred stimulus. So the different pairs of stimuli we're going to look at are simple patterns, complex patterns, simple objects, junk objects. So objects would be 3-dimensional, patterns would be 2-dimensional. Then we'll also look at natural scenes, conspecific faces. Conspecific faces would be cat faces, because they're the same as the species being studied, or human faces. Let's look at examples of all these so you can see what we're talking about. Oh, excuse me. First off, basically what we found was the big difference in processing between the congenitally deaf animals and the hearing animals was in the processing of faces. And specifically the learning of faces, conspecific faces and human faces. We didn't see any difference in the learning or recall of the other 5 different classes of visual discriminations that we looked at. Okay, so these other discriminations were patterns. So you can see some of the examples of patterns that were tested at the top left. So you can see like a cross versus a circle, or a triangle versus an inverted triangle, and so forth. These would be simple 2-dimensional patterns. To make them more complex, if you will, would be the line just below that, where either a surround or some sort of overlying pattern, like in letter D of the lower line, shows the X's over the circle and the square, just to make the pattern a little bit more difficult. These would be complex patterns. Simple objects are what you see in the upper left. So these are 3-dimensional objects. In this case, these are children's play blocks, glued together in different shapes and painted flat black, or junk objects a la Mishkin, which are basically just things you find around the lab, and having the animal discriminate 2 different objects. And these would presumably be objects that they would never have seen before. And then finally, natural scenes, as you can see, things you get from like calendars and so forth, would also be scenes that laboratory animals would probably never have seen before. Okay, so let's look here first off at the patterns, or basically the animal's ability to learn to discriminate simple patterns in the upper line and complex patterns in the lower line. What you're going to see here is that the results are expressed in what we refer to as errors to criterion. Basically, the animals were presented with the 2 patterns. One was rewarded, one was not, and we're looking at how quickly they learned to discriminate the 2. And what we did was we counted the number of errors, or the mistakes that they made, in order on their way to achieving criterion performance, which was 92% on 2 consecutive days. So once the animals reached 92% on 2 consecutive days, we had deemed that they reached criterion performance, and then basically just counted up the errors or the mistakes they made. And what you can see here is for the height of each bar is mean areas to criterion for both the hearing and the deaf groups. The actual circles that you see are performance of individual animals in each group. So it's not standard error or standard deviation, but actual performance of those animals. What you can see, the take-home message is from these two types of patterns, is there was no difference in the ability of the deaf or the hearing animals to learn to discriminate either simple patterns or complex patterns. Okay, but here what you see, but we found that things were very, very different for faces. Okay, so here are two examples of conspecific faces that the animals had to learn to discriminate. In this case, these are two different gray cats. If you're not a cat person, they probably look the same to you. But if you are a cat person, you probably can readily identify that they're very different animals. And then down on the bottom would be faces of two Caucasian males with similar colored hair and similar facial features that the animals had to learn to discriminate. Okay, so here what you find, though, is when we look at the ability of the animals to learn faces, the upper line is conspecific faces and lower is human faces. But it doesn't really matter whether it was conspecific or human. What you actually find is that the deaf animals made fewer errors to criterion than the hearing animals, and in many cases, just half as many. So if you look at some of the examples, particularly from the upper line, errors to criterion for the deaf animals could be around 20, where the errors to criterion for the hearing animals would be closer to 40. And this difference was significant. But it did not matter whether it was them discriminating cat faces they'd never seen before or human faces they'd never seen before. The difference was the same. Okay, but we didn't find any difference on the ability of the animals to recall a previously learned facial discrimination, discrimination of any kind. So you see patterns at the top, then into objects, natural scenes, conspecific faces. What you see here is data that was collected 24 hours after they learned to discriminate a face and were presented with a single block of 25 trials. So this is percent correct for that block of 25 trials. But the take-home message here is that there was no difference in the ability of the deaf animals or the hearing animals to remember faces that they'd previously learned to discriminate. So there was no advantage for the deaf animals in remembering faces they learned to discriminate, but there was an advantage of the learning of them. Okay, so to summarize this aim for this study, the congenitally deaf cats have learning and recall abilities similar to hearing cats on most of the discriminations we tested: simple and complex patterns, simple and junk objects, and natural scenes. However, the congenitally deaf cats have learning but not recall abilities superior to the hearing cats on conspecific faces and on human faces. So there clearly seems to be something special about faces. So in the second aim, we're going to determine what role deaf auditory cortex may play in mediating these superior cognitive functions, or specifically the ability to learn new faces. To do that, we're going to deactivate specific sites in deaf auditory cortex while the deaf cats perform the visual discriminations for which they possess enhanced cognitive abilities. So once again, we're going to look at these four areas of the presumptive ventral or what processing stream. Here you can see thermal reconstructions of the regions that were deactivated. You can see the location of four cooling loops in the upper left-hand panel, and you can see obviously the exact same thermal scale we were looking at before and how well localized the actual individual deactivations are, both for A1, for A2, for area T, and for area IN. Okay, so this slide is just to remind you what the normal pattern looks like for the face discriminations. We actually saw this slide a little while ago, and you can see that the normal pattern for face discriminations, the deaf cats have about half as many errors to criterion as the hearing cats. So just kind of remember this pattern as normal. Okay, so the first thing we're going to do is we're going to deactivate all four areas while the animals learn new conspecific and human face discriminations. And when we do this, what you see is that the normal pattern isn't identified any longer. And in fact, the deaf animals now have as many errors to criterion as the hearing animals. So when we deactivate all four of these sites, we see that the advantage that the deaf animals has went away. Remember, this is the advantage that the deaf animals have normally, but when we deactivate all four areas together, the advantage completely disappears. And this is obviously, this is both for conspecific and for human faces. Now we actually want to look at what are the contributions of each one of these four areas individually. Okay, So here in the upper line, you see performance of learning conspecific faces during bilateral deactivation of A1. Below that is the learning of conspecific faces during bilateral deactivation of A2, and then bilateral deactivation of area T, and the bottom line is bilateral deactivation of area IN. And what you can see is for A and A1 that it seems to be the normal pattern of learning. The deaf animals are better than the hearing animals, or they have fewer errors to criterion. And similarly, you see that on the bottom line for IN. However, for this area T, on the third line from the top, you see that the advantage has gone away. Okay, there is no more performance advantage for these animals when you bilaterally deactivate area T. And the other areas don't seem to be making little, if any, contribution to this enhanced face discrimination learning. Now, this is what we saw with conspecifics. We see the exact same thing with human faces. The upper two lines are area A1 and area A2, and performance there seems to be pretty normal, with the deaf animals having an advantage over the hearing animals. Similarly, in the lower line for area IN, you can see the deaf animals have an advantage over the hearing animals. But once again, for the human face discrimination learning, you see that bilateral deactivation of area T means that there is no performance advantage for the deaf animals. The advantage goes away. Now, a unique aspect of these experiments is that we can not only do bilateral deactivations, but we also do unilateral deactivations to see if a function is lateralized to one hemisphere or the other. On the top line, you can see bilateral deactivation of area T for conspecific face learning, and you see the advantage goes away. The second line is unilateral deactivation of area T in the left hemisphere, and you can see that there is no performance advantage for the deaf animals. And the bottom line is unilateral deactivation of area T in the right hemisphere. And here you can see that the advantage persists. So what this seems to indicate is that it's specifically left area T that is essential or performing an essential function for the enhanced learning of faces in the deaf animals, and it's not right area T. So why area T? What do we actually know about area T in hearing animals? Well, in previous work we've done looking at discriminations of different tones, noises, and vocalizations. In this case, since they're cats, they would be meows. And cats can discriminate each other based on their meow. What you find is that in hearing animals, bilateral deactivation of area T results in conspecific vocalization deficits. That means they lose the ability to discriminate each other based on vocalizations. The unilateral deactivation of left but not right area T causes deficits during conspecific vocalization discriminations. So you see in the right-hand group, or the right histogram, warm performance is in dark green. The lightest performance is bilateral, and just to the left of that is left unilateral deactivation, and just to the right of that is right unilateral deactivation. So you see there is no difference with right unilateral deactivation, but there is a difference, not to chance levels, but there is a significant drop in performance during left area T discrimination of vocalizations. So that is, in a normal hearing animal, area T makes a critical contribution to vocalization discrimination, specifically in the left hemisphere. So to summarize this part, we find that bilateral deactivation of area T eliminates the faster learning of conspecific in human faces identified in deaf compared to hearing cats. These enhanced face learning abilities were further localized to area T of the left hemisphere. Third, that left area T in hearing animals is critical for discriminating conspecifics acoustically via vocalizations, while left area T in deaf cats is critical for discriminating conspecifics visually via faces. Finally, cross-modal plasticity identified in area T reveals a cortical region that may be critical in discriminating individuals. Okay, so let's summarize the results from these two studies. In the first study, we saw we examined the contributions of PAF and the dorsal zone. In the final study, we looked at the contributions of area T. And we're going to look at the contributions that are normally made by these areas in hearing animals and in deaf animals, and then what's the common behavior between the two. So when we consider the posterior auditory field, in hearing animals, it normally makes a contribution to acoustic localization. In deaf animals, it normally makes a contribution to visual localization. So the common behavior between the two is localization. For the dorsal zone, we saw that in hearing animals, it makes an important contribution to acoustic motion processing. In the deaf animals, it makes an important contribution to visual motion processing. Therefore, motion processing is the common behavior between the two different conditions. And then finally, for area T, in hearing animals, we found that area T makes an important contribution to localization discrimination, and in the deaf animals, it makes an important contribution to face discrimination. So here we propose that the common behavior is the ability to discriminate individuals, either visually or acoustically. Okay, so the idea here is that cross-modal reorganization in the congenitally deaf switches the sensory but not the behavioral roles of auditory cortex. So they're common behaviors that are present regardless of whether there's normal auditory input or not. So the idea here is, if you look at this a brain that we saw earlier in the presentation, this is a lateral view of a cat brain. The yellow areas are auditory cortex and normally are making contributions to auditory behaviors. But following hearing loss in the deaf animal, what you find is there are particular areas that are now making contributions to visual processing, and there are other areas making contributions to somatosensory processing, which we didn't cover today. So we saw that the dorsal zone, the posterior auditory field, and area T are all visual areas from what we saw today. The field of the anterior ectosylvian sulcus, abbreviated FAS, also makes important contributions to vision during deafness, and the anterior auditory field makes important contributions to somatosensation processes following deafness. So hopefully what you remember from today's presentation is that congenitally deaf cats have specific visual abilities that are superior to hearing cats, and these enhancements are really no different from that of human subjects. Second, regions of deaf auditory cortex are responsible for mediating the enhanced visual abilities of the congenitally deaf. So, in the absence of normal auditory input, other senses such as vision and somatosensation do utilize the underpurposed auditory cortex in a deaf subject. And finally, cross-modal reorganization of the congenitally deaf switches the sensory but not the behavioral roles of auditory cortex. I'd like to thank all of our funding sources for helping us accomplish these studies. I'd also like to thank Professor Alex Meredith, shown on the left, a collaborator at Virginia Commonwealth University in the States, and Professor Andre Kroll, a longtime collaborator who's at the Medical University of Hanover in Germany, who's pictured on the right. I also need to thank all the people doing all the heavy lifting on these studies in the lab, specifically Nicole Chabot, Blake Butler, Carmen Wong, Dan Stolzberg, Tricia Brown, Melanie Cook. If you'd like to get any other information or read more about these studies, our website is cerebralsystems.ca, and our address and email address is provided on this slide. Thank you very much.
Thank you, Dr. Lomber, for that wonderful presentation. We appreciate it. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question drop-down box located on the far left of your presentation window. Type that question into the box that appears on the screen and click the Send button. We'll answer as many as we have time for during the end of the presentation here. So let's take a look at our current questions already coming in from our audience members. Our first question is, do the inputs to auditory cortex change following in the deaf?
This is a great question. Obviously, as you mentioned before, at the end of all these experiments, in addition to electrophysiological recording, we're also doing pathway tracer injections into these areas so that we can ascertain if there's been any changes in inputs and outputs to these areas following the loss of hearing. So the interesting thing is our original hypothesis was that there would be larger inputs from visual parts of the brain following hearing loss. And surprisingly, we're not really finding strong changes in the overall pattern of inputs that you would normally see to auditory cortex. Surprisingly, the inputs seem to come from the same constellations of cortical and thalamic areas that you'd expect. However, we are seeing some quite interesting changes in the outputs from auditory cortex, which are seem to be much more significant than changes in the inputs. Specifically, we've been looking at changes to tectal structures like the superior colliculus and the inferior colliculus, and we do see quite profound changes in the outputs from auditory cortex following hearing loss, but not really big changes in the inputs to auditory cortex. That was a great question.
Thank you. And here's our next question, Dr. Lomber. Why do other senses only take over certain regions of auditory cortex and not others?
This is also a good question. Our original hypothesis was that if auditory cortex in the deaf was going to be involved in mediating superior visual or somatosensory functions, that it would be distributed over multiple areas. And we were actually surprised to see how well localized it is to specific areas. And that this idea of an area having a particular behavioral function, like localization or motion or identity processing, and that basically that stays, and then you just substitute a different modality in for that same function. So we were surprised that things were that well localized. We were expecting things to be much more distributed. And also, I mean, we're expecting that we may find this with other tasks. We just haven't gotten to them yet. Thanks.
Thank you very much. And I want to also thank our audience members for their great questions coming in, and remind them that any questions not answered today will be answered via email. Here's our third question. How long can the brain be deactivated with cooling?
The longest we've had a region deactivated with cooling was about 2 hours. For most of our behavioral work, a region would be deactivated generally for, say, 20 or 30 minutes. But 2 hours on the outside. We really haven't done any experiments to see what's the outer window for reversibility. That is, you know, how long can we have an area deactivated and then have everything return completely to normal? But we really don't have any experiments that would require us to have a region deactivated for more than 2 hours. I mean, that would be a really long time for us. But we found complete reversibility within that sort of time frame. So what the outer window might be is still unknown, but the window that we're working with is much larger than anything we would presently need. Thanks, that was a good question.
Thank you. Our next question from our audience is, do similar changes occur in visual cortex of the blind?
Yeah, that's a really good comparison. Yeah, a lot of, obviously there have been a lot of studies also done on visual cortex in blind individuals, and very similar changes are being found in blind individuals. Whether that's using human functional imaging studies, looking at activation in visual cortex of the blind, or looking at animal studies in blind animals as well. Very similar kind of, this idea of a task-specific organization to the brain is being found, and also similar enhancements to vision or somatosensory function, to audition or somatosensory function, have been found in individuals that are blind. So we find a lot of parallels between studies done in the deaf and studies done in the blind. That was a great question.
Thank you for that. And we have time for one more question from our audience, and here's an interesting one. Why conduct these studies on cats as opposed to rats or mice?
Oh, yeah, that's very important. We and others have found that the wiring of the brain of rodents is very different from that of higher mammals, such as monkeys, cats, ferrets. And we're not sure if it's just got to do with the sheer size of the brain, or if it's more fundamental. But in many cases, what's been identified in rodent brains is that there are direct connections between, say, primary visual cortex and primary auditory cortex, or primary somatosensory cortex and primary auditory cortex. And these connections just don't exist in higher order mammals. So if you really want to understand the consequences of, say, blindness or deafness, you'd probably want to do that in a model organism where the brain is wired more like a human brain, and not have unusual connections that aren't typical of more highly developed mammals. So that's why you find more of these studies done in mammals such as monkeys, cats, ferrets, as opposed to mice and rats. It's just that the brain is different. Thanks for that question.
Dr. Lomber, thank you again for your presentation and for your important research. And I'd also like to take the opportunity to thank LabRoots for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast will be available for on-demand viewing through June 2018. You'll receive an email from LabRoots letting you know when the webcast will be available for replay. Please share that announcement with your colleagues who may have not been able to join us today for this live event. That's all for now, and thank you for joining us. We hope to see you again soon. Bye-bye.